Hello, students. Let's start a new chapter today. And the name of the chapter is Cell Cycle and Cell Division. As you know, that all multicellular organisms start life as a single cell, that is, the fertilized egg or the zygote. And this zygote, it grows by addition of new cells. And finally, the embryo is formed. So these new cells, this arise by division of the pre-existing cell, that is the zygote. So how this zygote, it divides and forms a mass of cell, and what are the processes that takes place in this cell division process, so that we are going to learn in this chapter. So let's start with cell cycle. Now, what is cell cycle? Cell cycle is the sequence of events that leads to the duplication of genetic material of the cell, synthesis of cell substances, and division of the cell into daughter cells. So as you know that when a cell divides, it needs to form all the parts within the cell that we have studied in the chapter Cell Structure and its Function. So it needs to form all those structures within it, and therefore here the duplication of those parts, right from the duplication of the genetic material, that is the DNA, to RNA, to the synthesis of the cell substances within it, till the division of the cell is completed, is called as cell cycle. Or you can say that before cell division, the preparatory phase that a cell undergoes, that is only known as cell division. So in simple words, these are the sequence of events involving growth and division of a cell from the time of its formation to the formation of its own daughter cells. I hope you have understood the term cell cycle. So this is the preparatory phase. Now, what is the generation time, or what time or how much time does it require for a cell to get ready before a cell division? So generation time, this is the period between two successive cell divisions, and it depends on the type of the cell and the external factors such as temperature, food, oxygen supplies. As for example, for a bacterial cell, it takes around 20 minutes in order to divide. So its cell cycle, it will be 20 minutes. Say for example, if it is a epithelial cell of the intestine, then it takes around 8 to 10 hours. If it is an onion root tip cell, then it takes around 20 hours. And for us, for a human cell, it takes approximately every 24 hours for a cell cycle. That is the time from one successive cell division to the next successive cell division. So what you have found out that for different types of cells, the cell cycle, it will be different. Now similarly, what type of or when a cell undergoes cell cycle? So these are the properties of cell cycle, that it has attained full size, then cell have to be a mature one, then it has acquired necessary energy for undergoing division, then formation of nuclear membrane is complete. So without the formation of nuclear membrane in case of eukaryotic cell, there the cell cycle will not begin and the synthesis of DNA is complete. So only then the cell cycle will start. Now let's come to the important part, that is the phases of cell cycle. So for a typical eukaryotic somatic cell, so here I am going to talk about a somatic cell, that is our vegetative cell. And what are somatic cells? These are the cells in our body parts except for the reproductive cell. All the cells of the body, that is called a somatic cell or vegetative cell. So a typical eukaryotic somatic cell, it has two main phases. One is the interphase, which is the long and undividing state, or this is also called as the I phase, and the shorter phase of nuclear division, that is called as the mitotic or M phase. That means a cell cycle, it has two main phases. One is the interphase, where no cell division takes place, and the next is the mitotic phase. It is when a cell divides. And all this cycle, it is controlled by some proteins. Now let's discuss about the interphase in detail. So interphase, it is a period of synthesis and growth which involves a series of changes in a newly formed cell before a cell division. So this is the preparatory phase and interphase, that is the first part where all the synthesis of the materials takes place that are involved with a series of changes in a newly formed cell. That means just after a cell division, the cell, it enters into the interphase. And the cell produces different biological molecules such as carbohydrates, lipids, proteins, and the nucleic acids that are required for its growth and carrying out the specific functions. So before the cell division, as this mother cell, as we call it, this is the mother cell, and the mother cell requires to distribute its inner content equally to the daughter cells. So in the first place, just after cell division, the mother cell, it will start preparing all those materials within it. And this interphase it lasts for about 10 to 20 hours. And as I have said, that for a human cell, it takes around 24 hours. Now let's see what happens in this whole state of interphase, which is the undividing phase. So this interphase, it is further divided based on its duration into 3 periods. The first is G1 phase, then the second is S phase, and the third is G2 phase. G1 phase, that means gap 1 phase. So it is a stage of initial growth of a newly formed cell. Here, the formation of nucleotides. Nucleotide, as you know, that is a part of the DNA, and this nucleotide, these are the single units in the DNA molecule. ATP, amino acids, DNA replication, All this starts in the G1 phase. Multiplication of mitochondria, chloroplast, formation of endoplasmic reticulum, Golgi apparatus, and other cell organelles. This all takes place in this state. In the preparatory phase that we regarded as the interphase, so here in the G1 phase, the formation of all the raw materials will be required, and the formation of those raw materials such as nucleotides, amino acids, the different type of carbohydrates that are synthesized within the cell itself will start with the DNA replication and multiplication of all the necessary cell organelles within it. Generally, the G1 phase that is shorter, that is a shorter period, and right after the G1 phase, the S phase starts. The S phase, which means synthesis phase. So it is the stage of replication of each chromosome by synthesis of a new DNA molecule on the template of the existing DNA. So the DNA, there is a parental strand which we call it as the template, and from the template strand the new DNA molecules are formed. As the cell divides to form two new cells, similarly the DNA molecule that also gives rise to two daughter DNA molecule, and the one from which the DNA replication it begins, that is called as the template strand. So here in the S phase, the DNA replication it gets started and the sister chromatids are formed for the parental chromosome and are held together at the region of centromere, and histone proteins are synthesized with the DNA strand forming a nucleosome. So along with the cell organelles, as you know, that the DNA material also needs to be divided so that it could be transferred into the daughter cells equally. So from the parental DNA strand, here in this S phase, which means the synthesis phase, so here the sister chromatids will get formed, and along with that a particular protein which is called as the histone protein, so that will form in order to form the nucleosome. And this daughter DNA, it will remain held at the region of the centromere of the chromosome. Then comes the G2 phase. So G2 phase, so it is the stage of further growth of the cell where the synthesis of RNAs and proteins continue. So generally, the cell or the DNA content of the cell, it follows a rule which is called as the central dogma, where from the DNA molecule, the RNA molecule is formed, and from the RNA molecule, the protein molecules are formed. So as you know that in the S phase, a new daughter DNA molecule is formed. So from that, the synthesis of RNAs takes place, and from RNAs, different types of protein are synthesized. So in the G2 phase, such further growth of the cell takes place before the cell divides. And during stage, the cytoplasmic organelles such as centrioles, mitochondria, and the Golgi apparatus, they are completely doubled. As you know that in the G1 phase, the multiplication it starts, and till the G2 phase, the doubling of all the cell organelles that gets completed, and the membranes they are assembled, then the spindle fibers for the cell division process that is also formed, and active metabolism starts in the cell just before the cell division. This G2 phase, it lasts for about 2 to 5 hours, and just after G2 phase it starts the next phase, that is the mitotic phase. Mitotic phase, so this is the final phase of cell cycle, and in this phase the cell division it starts. The mitotic phase, it starts with the prophase, metaphase, anaphase, telophase, and cytokinesis. So these are the 4 different stages in which the division of the cell takes place, and finally the cell it get separated, and that step it is called as cytokinesis. So generally in this mitotic phase, at first the division of the nucleus takes place, so that we call it as the karyokinesis, and under karyokinesis comes this 4 stages. So that is prophase, metaphase, anaphase, and telophase. And when the division of the nucleus gets completed, which results into 2 nucleus, so finally the cell cytoplasm that divides, and that we call it to be cytokinesis. And at last comes G0 phase, or it is also called as the quiescent stage, and it is the stage of inactivation of cell cycle, and further division of a cell stops, and the cell remains metabolically active. So this is a resting period for a cell. We can say that to be a resting period where the cell again do not divide, and it do not proceed to the next interphase as well. That means that doesn't start the G1 phase. So it undergoes a period of rest, and this stage, therefore, it is called as the quiescent stage or G0 phase. But the cell, it remains metabolically active. And finally, when the time comes, again the G0 phase that will get alternated with the regular sequence, that is G1, S, and G2 phase of the interphase and M phase of the cell cycle, and together this regular sequence of a cell that is called as cell division. So therefore it is the period in between one cell cycle to another, one cell division to another cell division that a cell undergoes. So today you have known that how a cell prepares itself and undergoes cell division, and the period from one cell division to the next cell division, and this whole cycle that takes place within a cell, that in simple words is the cell cycle. Thank you.